Welcome to our presentation on the adrenergic neurohumoral transmission. This presentation will talk about the catecholamines. Now, what are catecholamines, their functions and their biosynthesis, and the role in the adrenergic neurohumoral transmission. We are also going to talk about the adrenergic receptors, their classifications, their functions, the agonist of the adrenergic receptors, the affinity of the different agonists to the adrenergic receptors, and the MOA of these agonists. So what are catecholamines? So when we say catecholamines, these refer to the norepinephrine or NE, epinephrine, and dopamine. So they are considered to be endogenous, meaning that they are produced within the body. They are the sympathetic, neural, and humoral transmitter substances in most mammalian species. So they are considered to be neurotransmitters, and they are usually classified under the sympathetic nervous system. The NE and dopamine are considered to be the neurotransmitters in the impulse transmission in the central nervous system. And the norepinephrine, on the other hand, is the neurotransmitter at most peripheral sympathetic neuroeffector junction. So again, when we say neuroeffector junction, that is a junction between the sympathetic postganglionic neuron and the target organ or the effector organ. So where are these catecholamines synthesized? So they are synthesized in the brain, in the adrenal medulla, and some sympathetic nerve fibers. So again, these neurotransmitters are important in the transmission of impulse or information from the postganglionic neurons, so from one neuron to the other, or from one neuron to a target organ or an effector organ. Epinephrine is the major hormone released from the adrenal medulla. So the other name of epinephrine is adrenaline. And of course, the epinephrine is derived from norepinephrine. This diagram shows the biosynthesis of the catecholamines, dopamine, norepinephrine, and epinephrine. So the synthesis of epinephrine and norepinephrine is regulated by catecholamine synthesizing enzymes. So along the way, there are different enzymes that are important for the formation of these catecholamines. So it starts with the conversion of the amino acid phenylalanine to tyrosine or L-tyrosine. So this is a step one in the formation of the catecholamines. Then after that, the tyrosine will be hydroxylated to form L-DOPA. The L-DOPA will be decarboxylated with a specific enzyme to form dopamine. So dopamine is a neurotransmitter. Then it is a catecholamine. So dopamine will again be hydroxylated to form norepinephrine. And the epinephrine will be formed from norepinephrine by addition of the methyl group to norepinephrine by, of course, we have the enzyme there that is important, and that is known as the phenylethanolamine and methyltransferase, or the PNMT. So the norepinephrine and epinephrine both act as a neurotransmitter for sympathetic innervation. So this is the summary of the formation of the catecholamines, dopamine, norepinephrine, and epinephrine. Let's now proceed to the adrenergic receptors. So when we say adrenergic receptors, these are the receptors that subserve the response of the sympathetic nervous system. So again, when we say sympathetic nervous system, that is the fight or flight response. It is divided into two discrete subtypes. We have the alpha adrenergic receptors and the beta adrenergic receptors. The adrenergic receptors are also known as adrenoceptors. These receptors are again classified into two. We have the alpha and the beta receptors. For the alpha receptors, it is subdivided into the alpha 1 adrenergic receptor or adrenoceptor and the alpha 2 adrenoceptors. For the beta 2, or rather beta receptors, it is subdivided into three. We have the beta 1, beta 2, and the beta 3 adrenoceptors. The alpha receptors are the receptors that mediate the vasoconstrictor actions of catecholamines. So again, we have the alpha 1 and the alpha 2 receptors. Both the epinephrine and norepinephrine are considered to be agonists of the alpha receptors, and they activate, so when we say agonist, they activate the alpha 1 and the alpha 2 receptors. The alpha receptors, just like any other adrenergic receptors, they are considered to be G protein coupled receptor. So they utilize G protein for their action. So these alpha 1 receptors, they are coupled with GQ proteins. So these GQ proteins are important to cause increased release of calcium from calcium storage structures in the cell, such as the endoplasmic reticulum. So the increased intracellular calcium will lead to the muscle contraction, or in the case of the blood vessel, so it will cause, for example, vasoconstriction. They are primarily located on the smooth muscle throughout the body. So some of the important structures having the alpha 1 receptors are the vasculature. So when we say vasculature, it refers to the blood vessels. We also have the iris dilator muscle of the eye, the prostate, the urethral sphincter, the pylorus of the stomach, and the anal sphincter. This diagram shows the MOA of the agonist, particularly norepinephrine in the alpha 1 adrenergic receptors. So the receptor agonist will activate the signal transduction pathway as seen in this figure. So the alpha adrenergic receptor, just like any other adrenergic receptors, are considered to be G protein coupled receptors. So these are 7-membrane transprotein now receptors because they are called a 7-transmembrane protein because they span or they penetrate the cell membrane 7 times. So this structure here is our norepinephrine. It is considered to be um, an agonist of our alpha 1 adrenergic receptor. So these receptors are present on the postsynaptic membrane of the effector organs. So the activation of the alpha 1 receptors will initiate a series of reactions through a G protein activation of the phospholipase C, resulting in the generation of the IP3. So IP3 stands for the inositol triphosphate, as well as the generation of the DAG or the diacylglycerol. No? And this IP3 will initiate the release of calcium into the cytosol, and the DAG turns on the other proteins within the cell. So this mechanism, so this activation of the phospholipase C to generate the IP3, diacylglycerol, and increase in the calcium ions is important to promote the response of the alpha 1 adrenergic receptor. So the most prominent effect of the alpha 1 adrenergic receptor activation is the vasoconstriction, particularly of the blood vessels. This diagram shows the MOA and the biological effects of the activation of the alpha receptors, particularly the alpha 1 receptors. So the alpha 1 receptor is considered to be a GQ protein coupled receptor. So it is a GPCR, and it promotes or it produces its effects by increasing the calcium release from the calcium storage structure. So that is one of the mechanism whereby it produces its effects. So basically, this will act on the smooth muscles. So it will induce smooth muscle contraction. So for example, in the blood vessel, it will cause the vasoconstriction. So of course, when there is vasoconstriction, there will be increase in the blood pressure. So in the pyloric sphincter of the stomach, so it will induce the contraction of the pyloric sphincter so that digestion would be inhibited. And for the urinary bladder, it will induce the contraction of the urinary sphincter. So again, it will reduce the urination. For the eyes, it will act on the eyes, and particularly on the iris dilator muscle to induce pupillary dilation or mydriasis. So this affects pupillary dilation as well as the smooth muscle contractions, vasoconstriction, we are important during a fight or flight response. Let's now proceed to the alpha-2 adrenergic receptors. So these receptors are located on the presynaptic nerve endings and on other cells. Example, the beta cells of the pancreas. So again, the beta cells of the pancreas are important for insulin production, and on certain vascular smooth muscle cells. So when we say vascular smooth muscle cells, so these are associated with the blood vessels. When a sympathetic adrenergic nerve is stimulated, they release norepinephrine in the synaptic cleft, and it will interact with the alpha-1 receptors of the target tissue or the effector organ. A portion of the released norepinephrine will circle back and react with the alpha-2 receptors on the presynaptic nerve endings or the neuronal membrane. So what will happen if the norepinephrine will bind to the alpha-2 receptors that are located in the presynaptic nerve ending? So the stimulation of the alpha-2 receptor will cause feedback inhibition so that the release of norepinephrine from the stimulated adrenergic neuron will be inhibited. So this inhibitory action will decrease further the output from the adrenergic neuron and serves as a local modulating mechanism for reducing sympathetic neuromediator output when there is a high sympathetic activity. So note that in this instance, these receptors are acting as inhibitory autoreceptors. So the receptors, particularly the alpha-2 receptors, are considered to be inhibitory in nature. It inhibits its own secretion of the norepinephrine, in this case, the norepinephrine. So the alpha adrenergic receptors are also found on the presynaptic parasympathetic neurons. So again, when we say presynaptic neuron, that is a neuron that releases acetylcholine on its synapse. Norepinephrine released from a postsynaptic. So this should be postsynaptic sympathetic neuron, and it can diffuse to and interact with these receptors, the alpha-2 receptors, inhibiting the release of acetylcholine. So of course, now the postsynaptic sympathetic neuron will always release norepinephrine, and for those regions of the presynaptic parasympathetic neurons having the alpha-2 receptors, so when the norepinephrine will be able to bind to these receptors, it will cause the inhibition of the acetylcholine release. So this is another local modulating mechanism to control the autonomic activity in a given area. So in terms of the signal transduction pathway, in the effects of the binding to the alpha-2 receptors are mediated by inhibition of the adenylyl cyclase and a fall in the levels of CAMP. So the alpha-2 receptors are coupled with the GI proteins, and of course these are inhibitory, so they will decrease the CAMP levels. So in this example, we have here the postganglionic sympathetic neuron, so it releases norepinephrine in its synapse to act on the adrenergic receptors on the target cell. So when, for example, norepinephrine will bind to the alpha-2 receptors, so some of these are the norepinephrine that are released in the synapse. Some of these will attach or will circle back to the alpha-2 receptors that are located in the postganglionic sympathetic neuron. So binding of this to this alpha-2 receptors will decrease further the release of the norepinephrine. So this is a negative feedback inhibition. This is a form of autonomic modulation that is important for, so this is under notice that is important to modulate the sympathetic activity. Let's now proceed to the beta-adrenergic receptors. The beta-adrenergic receptors are mainly divided into three. So there are three types: the beta-1, beta-2, and the beta-3. So all the beta receptors are coupled with the GS proteins. So they are G protein coupled receptor. In this case, they utilize the GS protein, and this is important to increase the level of intracellular CAMP. Again, the CAMP serve as a second messenger in order for the effect of the beta receptors to be initiated. Where are these beta receptors mainly located? So some of the main locations for the beta 2 receptors include we have the lungs, we have the GIT, the bladder, the uterus, the pancreas, and the blood vessels. The beta receptors are mainly divided into three. Again, now we have the beta 1, beta 2, and beta 3. For the beta 1 receptors, they are mainly found in the heart and the kidneys. So in the heart, the binding of the agonist to the beta 1 receptors will increase the heart rate. So in the heart, again, in the heart, there are many beta 1 receptors. And when, for example, epinephrine, which is an agonist of the beta 1 receptor, will bind to this receptor, it can lead to increase in the HR or increase in the heart rate. It will also lead to increase in the contractility of the heart or increase in the stroke volume, which will increase both the cardiac output or the CO. And of course, when there is an increase in cardiac output, there will also be an increase in blood pressure. Meanwhile, on the kidney, the binding of the beta 1 receptors on the kidney, particularly on the juxtaglomerular cells of the kidneys, will cause the release of the enzyme renin. And of course, renin is important in the RAS system or the renin-angiotensin-aldosterone system. This RAS is important in order to increase the blood pressure. So of course, during a fight or flight response, we need to avoid hypovolemic shock, which is the depletion of the blood and the body fluids, so that the body tends to increase the blood pressure in order to also increase the volume of the blood that is present in the body. So when we are going to compare the potency of the different agonists to the beta receptors, so we have here three agonists of the beta receptors. We have the epinephrine, norepinephrine, and isoproterenol. So the isoproterenol is a synthetic agonist of the adrenergic receptor, and the epinephrine and norepinephrine are endogenous agonists. So when we are going to compare them, these are characterized by a strong response to isoproterenol with less sensitivity to epinephrine and norepinephrine. So it means that the isoproterenol is considered to be the most potent agonist in the beta receptors, while for the epinephrine and norepinephrine, the beta receptors have less sensitivity to these neurotransmitters. For the beta receptors, the rank order of potency of the different agonists is isoproterenol is the most potent, followed by epinephrine and norepinephrine. So the beta adrenergic adrenoceptors can again subdivide it into three, but in this case we only have two, because these are the major types of beta receptors: the beta 1, and beta 2. And of course, they can be subdivided based on their affinity for the adrenergic agonist and the antagonist. So again, when we say adrenergic agonist, it can refer to these. It could either be endogenous or synthetic. So isoproterenol is a synthetic beta receptor agonist, while these two are endogenous. So this slide will compare the two neurotransmitters in the sympathetic nervous system. You have the epinephrine and norepinephrine in terms of their affinity to the beta receptors, beta 1 and beta 2. For the beta 1 receptors, they have approximately equal affinities for epinephrine and norepinephrine. Meanwhile, for the beta 2 receptors, they have a higher affinity for epinephrine than from norepinephrine. So thus tissues with a predominance of beta 2 receptors. An example of a tissue with a predominant beta 2 receptors are the vasculature of the skeletal muscle. So they are particularly responsive to the hormonal effects of the circulating epinephrine that is released by the adrenal medulla. In terms of the signal transduction pathway, binding of the neurotransmitter at any beta receptors results in the activation of the adenyl and therefore increased concentrations of CAM within the cell. So this receptor is considered to be stimulatory, it utilizes the GS protein, and it will activate the adenylyl cyclase to increase the CAMP level. For the distribution of the receptors, so tissues such as the vasculature of the skeletal muscle have both the alpha 1 and the beta 2 receptors, but the beta 2 receptors predominate. For the heart, it contains predominantly the beta 1 receptors. These are the characteristic responses that are mediated by the different adrenoceptors. The stimulation of the alpha-1 receptors produces vasoconstriction, particularly in the skin and the abdominal viscera, and an increase in the total peripheral resistance and blood pressure. So of course, this is important. Now, peripheral vasoconstriction is important in order to divert the blood flow or the volume of the blood towards the vital organs such as the skeletal muscles and the heart during a fight or flight response. Conversely, the stimulation of the beta-1 receptors characteristically causes cardiac stimulation. So for example, it will increase the heart rate and the heart contraction, increasing the blood pressure. Whereas the stimulation of the beta-2 receptors will produce vasodilation in the skeletal vascular beds and broncholar relaxation. So again, the skeletal vascular beds are predominant. Now they have predominant beta-2 receptors. So this is important in order to supply the skeletal muscles with enough blood during a fight or flight response. This is also important in the lungs because it will induce bronchodilation in order to supply more air in the lungs, and of course more air, more oxygen towards the blood. This diagram shows the predominant effects of the adrenergic receptors as well as the location of these receptors. The alpha-1 receptors are mainly located in the peripheral blood vessels, and it will induce vasoconstriction of the peripheral blood vessels. The alpha-2 receptors are mainly located in the presynaptic neuron. It will regulate the release of neurotransmitters, epinephrine and norepinephrine, as well as acetylcholine. It will decrease the tone motility, and it will also decrease the secretions of the gastrointestinal tract. For the beta-1 receptor, again, it predominates in the heart. Now, it is in the myocardium of the heart. It will increase the heart rate, increase in the force of myocardial contraction. For the beta-2 receptors, these are mainly located on the peripheral blood vessels of the skeletal muscle and the bronchial smooth muscles. It will induce the vasodilation of the peripheral vessels in the skeletal muscle and bronchodilation. This diagram shows the potency of the different agonists to the different adrenergic receptors. For the alpha-1 receptors, the epinephrine is considered to be the most potent agonist, followed by norepinephrine, phenylephrine, PE, and the isoproterenol. So the isoproterenol is the weakest. For the alpha-2 receptors, norepinephrine is the most potent, followed by epinephrine, phenylephrine, and isoproterenol. So again, phenylephrine and isoproterenol are synthetic, and NE and epinephrine are endogenous. For the beta-1 receptor, of course, the most potent is isoproterenol, followed by epinephrine and norepinephrine. So they have equal effect. For the beta-2 receptors, the most potent is isoproterenol, followed by epinephrine and norepinephrine. So therefore, the epinephrine is, when we are going to compare these endogenous neurotransmitters, epinephrine and norepinephrine, for the alpha-1 receptor, so mas potent yung epinephrine compared to norepinephrine. For the alpha-2, mas potent yung norepinephrine as compared to epinephrine. For the beta-1, so they have the same effect. And for the beta-2, epinephrine is more potent than norepinephrine. In terms of the second messengers that they utilize, so this activation of the alpha-1 receptor will induce the production of the IP3, calcium, and the DAG. And in terms of the G protein utilized, it would utilize the GQ protein. For the alpha-2 receptor, so it will, since this is an inhibitory receptor, utilizing the GI protein, it will decrease the CAMP level. And for the beta 1 and the beta 2 receptors, so these are stimulatory in nature, they will increase the intracellular CAMP and they will utilize the G protein, GS protein. So all of these receptors are GPCR or G protein coupled receptor. This is summary of the different adrenergic receptors and their effects, as well as the potency of the agonist, norepinephrine and epinephrine. So the alpha-1 receptor, so when we are going to activate the alpha-1 receptor, it will mainly induce vasoconstriction. So when there is vasoconstriction, it will increase blood pressure, it can cause mydriasis, and it can cause the closure of the bladder sphincter in order to inhibit urination. So this is true, no? And this happens during a fight or flight response. Of course, for the alpha-2 receptors, these are inhibitory in nature, so binding of the agonist to the alpha-2 receptors will inhibit the release of norepinephrine, acetylcholine, as well as inhibit the release of insulin from the pancreas. Activation of the beta-1 receptors, so again, the beta-1 receptors are mainly found in the heart. It will increase the heart rate, so for other sites, it will increase lipolysis. Again, for the heart, it will also increase the myocardial contractility, and for the kidney, it will increase renin. So, of course, renin is important for the RAS system in order to increase the blood pressure. So, for the beta-2 receptors, again, they are predominant in the vasculature bed of the skeletal muscle and the bronchiolar smooth muscle. So, this will induce vasodilation, decrease in the peripheral resistance, bronchodilation, glycogenolysis. So of course, this is important in order to break down glycogen into glucose to supply the needed energy in the body. So it will also increase the glucagon release. Of course, glucagon has an opposite effect to that of insulin. It will release glucose from the sites such as liver. It will also relax the uterine smooth muscles. So all of these effects are seen during a fight or flight response. In terms of the potency of norepinephrine to epinephrine in the adrenergic receptor, so for the alpha-1 receptor, mas potent yung norepinephrine than epinephrine. For the alpha-2, mas potent naman yung epinephrine. For the beta-1, they have the same effect. And for the beta-2, much potent yung epinephrine than norepinephrine. This diagram shows the effects of norepinephrine and epinephrine in different organs having different concentrations or different quantity of the adrenergic receptors. So of course, in the vascular smooth muscles, there is a large number of alpha-1 receptors compared to the beta-2 receptors. So for example, when we are going to activate the alpha-1 receptors, it will induce the contraction of the blood vessels. Activation of the beta-2 receptors, on the other hand, will induce the relaxation of the blood vessels and bronchodilation. So at low epinephrine concentrations, the beta-2 adrenergic receptors will be occupied because these receptors have higher affinity for epinephrine. So we have here our beta-2 receptors. It is located for example, in the lungs, in the bronchus. And we also have here our alpha-1 receptors. So yung epinephrine, when we are going to compare yung dalawa, beta-2 at saka alpha-1, it has more affinity to beta-2 than alpha-1. So ibig sabihin at low epinephrine concentrations, mas mag-bind yung epinephrine sa beta-2 because it has higher affinity for that receptor. So ibig sabihin, when it occupies the receptors of beta-2, it will now subserve the function of the beta-2, which is relaxation of the bronchioles and the blood vessels. However, at high epinephrine concentrations, the alpha-1 adrenergic receptor would also be occupied because there are more of these receptors. The predominant effect at the high epinephrine concentration is vascular smooth muscle contraction. So again, when we are going to compare the number of the receptors, beta 2 and alpha 1 receptors, so mas madami yung alpha 1 kaysa beta 2 receptors. So pag mas madami naman, mas mataas yung level ng epinephrine, although yung epinephrine mas meron siyang affinity sa beta 2 at kaysa sa alpha 1, but of course in higher epinephrine concentrations, after occupying the beta 2, mag-occupy din siya ngayon sa alpha 1 receptors. Now, yung excess dito will also occupy the alpha 1 receptors. So when this happens, the predominant effect would be the effect of the receptor having more quantity. So in this case, yung alpha 1 receptors, mas madami siya compared to the beta 2 receptors. That is why yung predominant effect that will be predominant in this type of reaction would be the contraction. Now, because more of the epinephrine has bound to the alpha 1 receptors compared to the beta 2 receptors.